Lactate dehydrogenase is an extremely commonly looked at marker for cancer. And so if there's elevated lactate dehydrogenase activity in the blood, it's suggestive of cancer. And if you read, and I hope you read by now, the lactate dehydrogenase as the 11th step of glycolysis, well, here it is. In a normal cell, so we'll say healthy cell, or I should say non-cancerous cell, we're following the standard aerobic respiration approach to making ATP. So we have glycolysis, right? It's shown here. We have conversion of pyruvate to acetyl-CoA, and that's accomplished by the pyruvate dehydrogenase complex. Then we have TCA cycle, electron transport chain, ATP synthase, and we get tons and tons of ATP. Lactate dehydrogenase is still active in normal healthy cells, but we have this low level of activity here, and essentially we can have conversion of lactate back to pyruvate. And so there's a very, very low activity here, right? Low lactate dehydrogenase activity, and there's essential reversibility here. In a cancer cell, however, we have very highly increased lactate dehydrogenase activity. So now essentially this reaction is moving in the direction of formation of lactate, and then lactate is overwhelming the cell. And you can see here we have a lactate transporter. And so lactate is able to pass out of the cell. Lactate is going to acidify, so increasing the hydrogen ion concentration, which is essentially measured as a decrease in pH. So we increase the acidity of the interstitial environment, so of the extracellular fluid, right, or the tissue fluid. And this acid enriched environment selects for acid tolerant cancer cells. And so again, cancer cells have an advantage, right? Advantage. And so the healthy non-cancerous cells are harmed, and they're not going to thrive in this acid-rich environment. So again, there are benefits to cancer cells in formation of lactate, because with this enhanced production of lactate, we're increasing the amount of NAD plus that's regenerated to continue running glycolysis, and lactate in the close extracellular environment of the cancer cells acidifies the extracellular environment, which is going to damage the ability of the healthy cells in order to thrive. And so by killing off the healthy cells, we're increasing the real estate, increasing the resources that are available exclusively to the cancer cells. Possible reasons for this Warburg effect, there are four main areas here. And if we look at these four areas, you can see that we have mitochondrial defects as the first area listed. Now, the mitochondria of cancer cells are pretty damaged. Let's put it that way. If we look at these mitochondria, they are struggling to deal with reactive oxygen species, ROS. And these are damaging the mitochondria. And again, the ETC is not running in these cancer cells. And the ETC is going to essentially be detoxifying oxygen at complex 4 as we have conversion then to water.
This isn't happening. And so reactive oxygen species build up in these mitochondria and they cause further damage to the mitochondria. Consider also that the mitochondrial genome, right, mitochondrial DNA contains 13 coding sequences for the protein chains which form the functional complexes of the electron transport chain. So not only are we not running the electron transport chain, but we can have also decreased ability to produce those complexes. So there are problems with the mitochondria within the cancer cells, right? And so that the genes, right, these 13 coding sequences which contribute to the structural and functional proteins that form the complexes of the respiratory chain, they're also being damaged. And there's a very high mutation rate in the mitochondrial chromosome, and there's not a lot of access to DNA repair. So mitochondria and cancer cells are damaged, and they're going to be further damaged, right? So we're not running the electron transport chain. And by not running the electron transport chain, we are contributing to this Warburg effect. So this is increased glycolysis, increased lactic acid dehydrogenase or lactate dehydrogenase, and we're also experiencing decreased TCA and decreased ETC. Hypoxia, of course, no oxygen. Well, how can you run the respiratory chain if you don't have molecular oxygen available for complex 4? And if we shut down any one complex of the respiratory chain, we shut down essentially the entire respiratory chain. We don't make the hydrogen ion gradient, and we don't run ATP synthase. As far as oncogenic signals are concerned, um, you're looking here at a number of cell signal proteins. And these proteins participate in phosphorylation cascades within cells. And you should know that insulin, again, is insulin carcinogenic? It's something that I think we can think about and argue. Insulin is going to drive activation of phosphatidylinositol triphosphate and ACT, which is kinase in this signaling pathway, and ultimately drive activation of protein kinase B. And so, anyway, insulin is driving activation of these factors, which are going to further activate glycolysis. There are also, last point here, altered metabolic enzymes. And so we have increased expression of hexokinase 2, which we did look at. And this increased expression of hexokinase 2 is going to drive glycolysis. We have increased expression of this TKTL1. This is trans ketolase that is important for the pentose phosphate pathway. And by increasing the expression of trans ketolase, we can increase the amount of ribose 5-phosphate, which is then available for nucleotide synthesis. So we support then the DNA replication, which is necessary for cell reproduction. And then there are mutation, right? See this? FH and SDH mutation. This is saying fumarate hydratase, fumarase, right? And so fumarate dehydrogenase is giving us then succinate, right, in the TCA cycle. And succinate dehydrogenase, you know, has 1 foot in the TCA cycle and 1 foot in the ETC. And so if we have mutation and depressed activity of
fumarase and also succinate dehydrogenase, then that's leading to decreased TCA flux and decreased electron transport chain flux. So again, we're driving then reliance of the cell on glycolysis and lactic acid fermentation. So to kind of sum this all up, there are a number of drugs which can be used in cancer treatment which target these enzymes in glycolysis and also these enzymes that we've looked at here in pentose phosphate pathway. So these drugs are targeting enzymes important in pentose phosphate pathway. And again, one of the products of this non-oxidative branch of pentose phosphate pathway is glyceraldehyde 3-phosphate, which then can flow into the payoff phase of glycolysis. And here are the drugs which are targeting hexokinase and also glyceraldehyde 3-phosphate dehydrogenase. So these drugs are useful, and there's kind of a benefit here of using drugs to hit glycolysis enzymes in cancer therapy because cancer cells are so highly dependent upon the glycolysis pathway. These drugs should have a good selective toxicity in damaging more so the cancer cells and not causing too much collateral damage to the healthy cells. And that's it, right? That's it. So we can leverage this altered approach to the central core of metabolism in cancer cells as a way to design drugs that have a good activity against cancer cells. And this is even more effective and more important because cancer cells in hypoxic environments are more resistant to traditional chemotherapy and radiation. So there's this, additional benefit of using drugs to target the glycolytic enzymes.